All right, I think we're going to go ahead and get started. Some of you who have been part of these webinars before will recognize that I am not Paul Gross. I am Michael Kruer, the presenter this evening. Paul actually is on a well-deserved vacation, getting some time away with his family, and so he asked me if I would be willing to introduce myself, which I'm happy to do. For those of you who have not participated in these webinars before, the MyCP webinar series really is designed to provide community accessible and meaningful information about the research that's being conducted by members of the CP Research Network. And so each month there's a principal investigator associated with one of the projects that the CPRN is engaged in. This month, it was my turn to share with you our work on the genetics of CP. And so I have a number of slides that I'd like to share with all of you. Please hold your questions to the end, although the presentation itself should only take roughly half of the time, unless there's some burning question or something that is not clear to you, then feel free to interrupt. Preferably by raising your hand, and then I can call on you, just given the number of participants that we have. So if that sounds good, I think I'm going to go ahead and get started. So again, my name is Michael Kruer. I'm a pediatric movement disorders neurologist working in Phoenix, and I also do a considerable amount of neurogenetics work, both in the clinic and in my research lab. So in terms of our agenda tonight, really it's several-fold. We wanted to just briefly review some of the aspects of the CPRN and also give an overview of MyCP. Then I'm going to launch into my discussion of the genetics of cerebral palsy and really try to focus in on practical aspects of some of the latest research findings. Then, as I mentioned, I'd like to use the last half of the time for questions and answers. So look forward to hearing from all of you. So the CP Research Network is really geared towards improving outcomes. And a huge focus of this is doing work that people with cerebral palsy value most through a combination of high quality clinical research, as well as quality initiatives. The value aspect of this is something that's very core to what we're trying to accomplish within the CPRN. And this was really set through a series of meetings and workshops back in 2017, and then ultimately published in 2018 under the name of Research CP. So as far as what CPRN actually is, it's really a collaborative network comprised of institutions across North America. It includes clinicians, therapists, researchers, patient advocates, individuals with CP, their families, all kind of coming together to conduct research to improve outcomes for people with cerebral palsy. There's a data coordinating center that's located at the University of Utah and overseen by Jacob Keene at that university. And CPRN is fundamentally both a clinical registry that gathers data while physicians and therapists are treating patients with CP. And so it gathers data on the patients themselves, on interventions, and how those interventions affect those individuals. And it does this through using an elaborate set of information gathering coming from electronic medical records. In addition, CPRN is a community registry that follows both kids and adults with CP over the long term. So then what is MyCP? MyCP essentially is a web portal arm that's owned and operated by CPRN and really intended for the extended CP community. MyCP offers participants the opportunity to participate in the community registry through surveys. It allows individuals to engage in discussions about research, to ask questions, and compare experiences. It shares news about CPRN studies, and it also provides tools for enhancing care. So for those of you who have not checked it out before, just encourage all of you to take a look at mycp.org and sign up if you're interested. So in terms of the overall structure of the network, there are a number of projects, as you can tell, that are at various stages of development and implementation. The goal of the network is really to accelerate research by providing tools in the form of the registries, as well as meetings among stakeholders in the process to try to facilitate collaborations. The registry is intended for research, but it's also intended to bring about improvements in care through quality improvement initiatives. And these two are really very much intertwined, incorporating adult care, dystonia diagnosis and treatment, hip surveillance, and a protocol to reduce infections for intrathecal baclofen pumps most recently. So the number of centers that are participating in the CPRN network is growing. As you can see, the centers that are depicted with the green dots are actively enrolling patients. There are a number of those sites, but in addition, there are a large number of centers that are getting very close. They're preparing their registry and hope to be enrolling soon. Still, in addition, there are several sites that are either candidate sites or are at the compliance and information technology stage where they're trying to do some of those challenges. All right, so that's the CPRN in a nutshell. And at this point, I'd like to go ahead and start with the main event, if you will, cerebral palsy and genetics. And the question that I wanted to pose for tonight's webinar was what difference does it make? For myself and many clinicians and researchers working in the CP space, we want our work to be relevant. We want our work to matter to all of you who are affected by CP. And so this is the lens that we'll be looking at things through this evening. And so just to start off, if those of you in the audience could just indicate in the chat, what are some of the common causes of CP? Okay, prematurity. Prematurity is certainly an important cause of CP and it is one of the leading causes of CP. We know that children born premature have a higher risk of CP, and particularly if they have additional adverse events associated with premature birth, bleeding in the brain, for example, those can increase the risk substantially. Lack of oxygen. So this is a classic, well-known cause or contributor to CP, something that most in the CP community are familiar with, and something that a great deal of very good basic science work has been done about. What else? Infection. So an infection is another important contributor to CP, and is something that may in some cases be overlooked, but in most studies comes up as an important contributor to CP, particularly prenatal infections or so-called in utero infections. So good. So all of these are important factors to consider. But I'm going to share with you another potential factor that maybe many of you are less familiar with, and that's genetics. Before I do so, though, I want to make the case that the reason that we do research as part of the CPRN is that research is good medicine, fundamentally. And I think that for many clinicians, there are a lot of patients to see and only so many of us. And so we want to take good care of everyone who comes to us. We want to be able to address all the need that exists. But I would also argue that the best long-term success is going to come from having a plan as to how we're going to improve things, at least a general sense. And that a lot of research gains can be thought of in terms of having a balanced portfolio, having both short and long-term goals, some short-term wins, but then some perhaps longer-term investments that may pay significant dividends in the end. And so, with that in mind, we really haven't had new treatments in CP for decades. One of the foci of the CPRN is trying to maximize what we've already got, trying to essentially develop gold standards for care, and really recognizing that we're still trying to understand how best to use the tools that we have at hand, and who best to use them for as well. And I think research of this kind can have a significant impact in the near term. And so this is an important goal. However, I would also counter by saying that it doesn't change the fact that we still need more in the pipeline. Again, looking towards long-term solutions, there ultimately will come a time where if nothing new were to come about in terms of CP interventions and therapeutics, that we could potentially exhaust what we do have. And so not only repurposing existing treatments, perhaps developed for other disorders, but also developing completely new treatments. And just to think outside the box, these could be things like drugs, like devices, such as brain stimulators, for example, gene therapies, even molecular forms of medicine like CRISPR-based technologies, I think are potential therapies that could be considered. One of the difficulties, I think, is that our knowledge of CP has been inherently limited, at least in the past. Most of the treatments for CP that we have are focused on reducing symptoms, not the fundamental problem. And so I think to some extent, this has been perpetuated by this idea that CP is a wastebasket diagnosis. This is an idea that is prevalent in some of the medical communities. This paper was published more than a decade ago by Terry Sanger, one of the godfathers of the pediatric movement disorders neurology field. And in it, Dr. Sanger really argues quite eloquently that this is not the case, that CP is a valid construct intrinsically, but that it's important to understand that there are probably different ways to get to CP. I think that this proves to be very important in the end, because figuring out how to improve one person's CP might be different than the solution for another person's CP. So for example, if you compare this to epilepsy, seizure disorders are very complex, very challenging, and many individuals with CP will have both epilepsy and a movement disorder. In some ways, though, CP may be even more complex, because in epilepsy, the goal is to stop the seizure, whereas in CP care, our goals are even higher in some respect, and that we're trying to ultimately improve function for people with these symptoms. So the question comes up, well, why genetics? Well, genetics can provide an important roadmap for research, and genetics essentially can give us a place to start. It gives us a stepping off point and a solid ground for which to build from. And so that's what attracted me to genetics and CP in the first place. Somewhat remarkably, this is an informal graph, and we need better studies to be more certain about these numbers. But to follow up on what we had discussed just a few slides ago, prematurity is recognized to be an important contributor to CP, perhaps in as many as 40% of cases. Infection, also an important contributor. Stroke and lack of oxygen as well, although many people are surprised about current estimates for the number of, or the proportion of cases that lack of oxygen is implicated in. Current estimates actually suggest that genetic causes, misspellings in a single gene in many cases, may actually account for a surprisingly high proportion of CP cases. And so that begs the question, so genetics meaning what? Well, think of it this way. All of us have a genetic makeup, a blueprint that contains the instructions for our bodies to develop and to function. That blueprint, if you will, can be thought of as a cookbook for how to make an individual human being, our genome. That cookbook, in turn, has many recipes in it. Roughly 20,000 recipes, and those recipes are individual genes, each of which has a specific function in our bodies. Those genes are written in the DNA alphabet, and here's where it can get complicated, because if there is a misspelling in that DNA alphabet, we might be intending to bake a chocolate cake, for example. But if there's a typo, a mutation that leads to a misspelling or a missing page, for example, in the recipe book, that can substantially affect the way that the cake turns out. So imagine that there was a typo in your recipe for a cake, and you didn't know if you needed 1 egg or 6 eggs. That could make quite a difference. Same thing with genetic mutations. They can substantially affect the proteins that perform many of the functions that our bodies rely on. So when we're talking about genes and genetic risk factors, I think that many times we've thought of genetic risk factors as something that's going to tip the scales, that is still going to need something in the environment. So for example, a person or a child might be more at risk of being born premature, but that doesn't necessarily in and of itself tip the scales from health to disease or a disorder. In this case, though, we're focused really on genes with high effect size, genes that have a substantial effect on the function of that particular protein. And so these mutations are so-called single gene causes of disease. Now, there are many other contributions of genetics to CP, but for the purposes of our discussion, this is the one that's been most revealing in recent studies. So why would this matter? Getting back to the heart of the matter, really, it's because that if we can identify genetic causes of CP, this may have important research implications and may be important for the field in the long term. This could, to some extent, change some of the trajectory of current research in CP. It might give us a new place to start in some cases, or new insights. There are also potentially important short-term clinical implications. These include things like implications for diagnosis, implications for prevention, for treatment, and even for family planning based upon recurrence risk. In addition, there are interesting and potentially important human implications. Many parents who have a child with CP have thought at one time or another, maybe I shouldn't have blank in relation to their child's CP. Time and time again, we find that there was nothing that the parents could have done differently. But it's been my experience that in some cases, finding a genetic cause can lead to some closure, which can be important in and of itself. So we're very excited about some of our recent findings, and this comes from an international collaboration with colleagues in Australia, different parts of the United States, as well as China. This has been accepted for publication in the journal Nature Genetics, and it is soon to be in press. But really, our studies form the first, provide the first firm statistical evidence that genetic mutations lead to CP. So this has been something that's been suggested for quite some time, but these particular studies show robust statistical evidence that this is the case for a proportion of individuals with CP. Throughout this process, we discovered several new CP genes, which is important in and of itself, although each gene only corresponds to a relatively small proportion of patients. So there's not one single CP gene, but actually at the current time, we're estimating that hundreds of CP genes exist and that many of those still await discovery. This seems like it's an overwhelming prospect, especially with hundreds of different genes all affecting brain development in complex ways. But fortunately, we're starting to identify key pathways, ways in which these genes and their proteins intersect within the cell, within the brain. And these key pathways, it seems, are starting to tell us that they're important for the development of cell-to-cell connections in the brain and for forming functional brain circuits, which we've long known has been an important part of CP, at least for some individuals. So furthermore, subsequent studies that are currently in review have found that if we look at a large group of individuals that undergo gene sequencing, individuals that have cerebral palsy, we find that for approximately a quarter of them, we can identify a genetic cause of their CP. This is a high rate, and this is a rate that we actually expect may increase with time, particularly as additional genes are discovered. Quite interestingly, some of these individuals also have another cause, quote unquote, for their CP, meaning that some of these individuals were found to be oxygen deprived. Some of these individuals were born premature. And in some cases, it's turning out that there's not just a simple explanation. It might not be just genes or environment, but it might be the interaction between both that turns out to be crucially important. One thing that I'm perhaps the most excited about is the fact that nearly 10% of individuals who undergo genetic sequencing will have a finding that changes their care. So as a physician that cares for many kids and young adults that have CP, this means that in almost 10% of cases now, even though we're just scratching the surface, we're coming up with findings that allow us to look at things in an exciting new way. And in some cases, avoid potentially dangerous risk factors, and in others, actually employ new treatments prompted by this fresh understanding of the causes of a person's CP. So this brings up the question, well, what about diagnosis? Does this have implications for diagnosis? And I think a fair question is, should doctors be evaluating children suspected of having CP differently? The current practice parameters are outstanding, but they were developed nearly 15 years ago. And things have changed quite a bit since then. I think it's becoming increasingly difficult to know what the cause of an individual's CP is without extensive investigations. And so the diagnosis of CP itself is becoming more and more complicated, particularly for primary care physicians. And so some of the latest recommendations are suggesting that perhaps seeing a specialist might need to become a part of early CP diagnosis. As I mentioned, prevention is an exciting aspect of what these findings potentially mean. The old saying, an ounce of prevention is worth a pound of cure, certainly applies here. And the opportunity to prevent problems before they start, whether that be problems with the CP itself or sometimes in the other organ systems that can be involved, is crucially important. Finally, treatment. Now, when I say treatment, changes or innovations in treatment can take multiple forms. In some cases, this means reaching for the best treatments first, rather than finding those through trial and error, as we're used to doing in CP management. In other cases, this can mean repurposing or using off-the-shelf existing treatments that were intended for another condition, but now using it for CP. And although this isn't common at the current time, we expect that as our understanding of the genetic basis of CP improves, this will start to become more and more commonplace. I'm particularly interested in using genetics to try to predict responses to interventions. This can be things like surgeries, such as deep brain stimulation, or it could even be therapeutic interventions. But I think until we have the studies, we won't know whether genetics can guide us as to who might best respond to a given treatment. And then finally, probably the most ambitious but potentially most exciting aspect of all of this work is that it suggests that entirely new treatments might be possible, treatments that are aimed at the fundamental problem that causes the CP in the first place. There are important family planning implications. So most individuals that have a child with CP, most parents are counseled that their risk is low of having another child with CP, somewhere around 1%. And in the case of a common form of genetic inheritance, that of so-called de novo or spontaneous mutations, in those cases, often the father does not carry the gene, the mother does not carry the gene either, and the child only acquires the mutant gene through early development. We now know that there are roughly between 50 and 100 genetic changes that occur from one generation to another. So some of these things happen by chance. But in other cases, in some families, depending upon the genetic cause of their CP, the recurrence risk could be significantly higher. For some, 25 to even 50%. And so this is the sort of information that families have indicated many of them want to have. The sense of community is important, particularly in the context of MyCP in this discussion. But one of the questions that sometimes arises is whether an individual who has a genetic cause for their CP identified, whether they still have CP. And in fact, I think most clinicians would currently argue that it's certainly possible to identify with both the CP community and the rare disease community. That the diagnosis of CP, if that's made on solid ground, that doesn't change based upon a genetic finding. However, that genetic finding might explain certain unique aspects of that individual's health. And so connecting with others in the rare disease community, other families who are dealing with mutations in the same gene, can sometimes be uniquely informative for those folks. So identifying with both communities may in fact be the best answer. In terms of what all this has to do with the CPRN, one exciting initiative that I'm looking forward to telling you about is that the CPRN CP genetics study. So this is supported by a National Institutes of Health grant. And the plan for this study is to enroll 500 families and to compare the DNA sequence of the healthy parents to the affected individual with CP. In some cases, that'll be a child under 18, but adults in their 40s and 50s can participate as long as their parents are also available to provide a DNA sample. By analyzing their genomes and comparing what's different in the DNA of the parents who are healthy to the child with CP, we hope to be able to identify mutations that lead to symptoms. So because of the current state of things with COVID and the pandemic, we've had to significantly adapt our study plan. And we still intend to roll the study out through CPRN centers. We anticipate that we'll be able to do it this winter, but we're taking a different approach, and we're utilizing an electronic consenting app that you can use through your phone, your tablet, or your computer to participate in the study. So you won't need to speak in person with the study coordinator. You'll get the information that you need in an easily digestible format. You'll be able to go through the consent process at your own pace. And then once you complete that process, our research team will actually send sample collection kits, saliva collection tubes, to your home. You can simply spit in the tube, which serves as a source of DNA, and then return it through a prepackaged mailer. So for those of you interested in participating in this study, and for those of you who are willing to partner with us to help drive genetics forward in CP, please ask about the study at your local CPRN center. So in conclusion, the genetic causes of CP are substantial. We're still trying to understand and don't want to overhype these findings, but we're also excited because of the fresh look this has given us at the neurobiological basis of cerebral palsy. Genetic findings can impact individuals and their care now, but also may have implications for ongoing research. So for any given individual, their genetic findings may or may not impact their care. But as a whole, those discoveries are helping to move the field forward. And then finally, perhaps most importantly, CPRN will be rolling out a major genetic study in late 2020. So with that, I want to thank all of you for your attention. If you have any questions, please direct them to us. Our lab email is kruerlab@phoenixchildrens.com. I'd like to thank my wonderful colleagues within CP Now and CPRN, as well as all of the individuals in my lab that make this research possible, in collaboration with all of the wonderful folks around the world that are helping to move the needle in the genomics of CP. Thank you so much, and I'd be happy to take any questions that you have.